Hi everyone, welcome back to my YouTube channel, and today we're going to learn about classification of gram-positive organism. So this is the lab algorithm for classification of gram-positive organisms. So let's discuss how they are divided. So whenever we do this gram stain, the organisms which have very thick layer of peptidoglycan layer in their cell wall will retain crystal violet dye and known as gram-positive organisms. So, uh, the organisms that are gram-positive have thick layer of peptidoglycan layer that will retain the crystal violet color and known as gram-positive organisms. Basically, these gram-positive organisms have their shape some like this circle, and they are known as cocci. And there are very few organisms that are gram-positive but have the shape of these rods, and they are known as gram-positive bacilli. And very few of them resembles of that of fungi, and they are known as the branching filaments, but they are the gram-positive organisms. Some example of this branching filaments are nocardia and actinomyces. And most of the bacteria that are gram-positive have the shape of this cocci. So we have to differentiate this cocci on the basis of test which is known as catalase test. In this test, what we have to know is there is the enzyme known as catalase. So this catalase will degrade hydrogen peroxide into water and oxygen. So there will be presence of bubble. So in this test, what we're going to do is we will put hydrogen peroxide plus bacteria in a slide. If those organisms are catalase positive, then there will be presence of bubble. And if that organisms are catalase negative, then there will be absence of bubble. So the catalase positive organisms is Staphylococcus. Now we will do another test which is known as Coagulase test to differentiate the staph organisms. So in this Coagulase test, what we're going to do is we will add plasma and bacteria in one tube, and we will look for the fibrin clot is forming or not. So if there is presence of clumping, then that is called coagulase positive organism, and if there is no clumping, then the organisms are known as coagulase negative organisms. And further, this can be differentiated by a test known as novobiocin sensitivity test. So this catalase positive organisms is Staphylococcus aureus, and the remaining organisms is again divided on the basis of another test known as novobiocin sensitivity test. In this test, what we will going to do is there will be one agar in which there will be a lot of bacteria. So we will put this novobiocin disc in this agar. So if the organism is sensitive to this novobiocin, then there will be one clearance zone around this disc. So they are the organisms that are sensitive to this organism, and the example is Staph epidermidis. But if the organism is resistant to this novobiocin sensitivity test, then there will be this novobiocin disc, and the bacteria can grow up to the edge of this novobiocin disc. There will be presence of no any zone of clearance around the disc. So that is the novobiocin sensitivity is not present, and those resistant organisms example is Staph saprophyticus. So this is all about Staphylococcus group. Now we will learn about Streptococcus organisms. So the organism that was catalase negative. So these organisms are the Streptococcus. And very important differentiating factor between Streptococcus and Staphylococcus is Staphylococcus is present in a cluster, in a group, but this Streptococcus are present in a chain. So we will do another one test that are going to differentiate between this Streptococcus, that is hemolysis. So In the blood agar, we will put mammalian blood and see if there is presence of hemolysis or not. If there will be complete hemolysis, then those organisms are known as beta hemolytic organism. So there will be complete hemolysis. And the organisms that will cause partial hemolysis, those organisms are known as alpha hemolytic streptococci, and they will give color somewhat greenish color. And the organisms that will have be no hemolysis, those organisms are the gamma hemolytic streptococcus. Now again further we have to divide these organisms on the basis of antibiotic sensitivity test. So it's very easy to remember. Here is beta, so beta for B. So we will do bacitracin sensitivity test. So like in case of novobiacin, we will put bacitracin disc over agar where there will be bacteria. So if the organism is sensitive to it, then there will be a zone of clearance, and if the bacteria is not sensitive, then that bacteria will grow up to the edge of this disc. So these bacitracin sensitive organisms are group A beta hemolytic streptococci group, for example Streptococcus pyogenes, and the resistance one is the group B beta hemolytic streptococci, example is Streptococcus azalacti. Now in this alpha hemolytic, so we can remember by O. So the antibiotic we are going to use is optocin sensitivity test. Like in case of this bacitracin, if the organism is sensitive, then there will be the zone of clearance, and the organism for this is Streptococcus pneumoniae. And the organisms that are resistant to this and optocin sensitivity, they are the viridans group of Streptococcus. Likewise, there is this gamma hemolytic, there will be no any hemolysis, and here we will see if the bacteria can grow in high concentration of salt or not. So there will be presence of NaCl, and we will look for the growth of the bacteria. If there will be growth, then those organisms are enterococci. And if there will be no growth, then they are the non-enterococci. And example for this enterococci is Staph bovis. So this is the basic classification of gram-positive organism. So in this summary chart, we know this is the gram-positive. So if there is branching filaments which will resemble as fungi, then they are known as nocardia and actinomyces. And there are very few bacilli that are gram-positive. And the example for this is Listeria, Bacillus, Clostridium, and Corynebacterium. And rest all maximum number of cocci are the gram-positive organism, and we can differentiate them on the basis of this test catalyst. Here we will look for the bubble. If there is presence of bubble, then those organisms are Staphylococci. Staphylococci are present in this clusters form, and further we will do a test known as coagulase test. Here we will look for the clumping, if there is formation of fibrin clot or not, and if there is presence of fibrin clot, then that organism is coagulase positive. An example for this coagulase positive is Staphylococcus aureus, and for rest of the staph groups we will do a test that is novobiacin sensitivity test. If the organism is sensitive, then that organism is Staph epidermidis. There will be the clear zone of clearance, and if the organism is not sensitive, then that organism will be Staphylococcus saprophyticus. Now let's go and learn about Streptococcus. This is Streptococcus are catalyst negative, and the Streptococci are present in chain-like structures, and we will do hemolysis. So they can be further divided into alpha, gamma, and beta. If there will be complete hemolysis, then that organism is beta hemolysis, and we will do another test, that is bacitracin sensitivity test. If the organism is sensitive, then that is group A beta hemolytic streptococci. Example is Streptococcus pyogenes. And if there is resistance to this antibiotic, then those organisms are group B hemolytic. For this example is Streptococcus as a lacti, and another is alpha hemolysis. Here will be the partial hemolysis and they will impart green color, and we will further divide this by doing optogen sensitivity test. If this is sensitive, then organism is Streptococcus pneumoniae. If it is negative, then those organisms are very dense group of streptococci. Example is Streptococcus mutans. And another is the gamma hemolytic. There will be no hemolysis, and we will check if these organisms are going to be growth in this presence of high concentration of salt. So we will put 6.5% anesthesia and look for the growth of organism. If the organism will grow, then that are known as enterococci. For example, enterococci fecalis or enterococci fecum. And these, if the organisms are unable to grow in this high concentration of salt, then they are non-enterococci. Example is Staph bovis. So this is all for today. I hope you guys understood this classification. We will meet up in next video.